Hey guys, this is Mr. Vree. We're gonna do a quick lesson on translation to hopefully help you and prepare you for your lab on Friday, where we'll model this process along with transcription and mutations. So, quick recap from yesterday. We went over transcription, where DNA is turned into messenger RNA, and today we're going to look at how does that messenger RNA produce a polypeptide or a protein. A couple of quick reminders. Codons are 3 nucleotides on the messenger RNA that code for a specific protein, amino acid. And so if we look at this chart, GCA, for example, here on this codon chart, we would look at the first letter, G, down here at the bottom, C at the top, the second nucleotide, and then A, the third nucleotide. And so that brings us all the way over here. That is the codon for the amino acid alanine. You'll notice that there's some redundancy to this. There's only 20 amino acids, and I think it's 64 different possible sequences of the 4 letters of RNA. And so some of these are stop codons, and one of them is a start codon, AUG, but there's just some redundancy. Also, keep in mind that this is universal, so it doesn't matter if we're a human being or we're a bacteria. This is codon chart is used for all organisms. Watch a quick animation here of this process. I'm gonna be the talker. So what we're looking at here is a tRNA, a transfer RNA flying in, and that purple thing is a ribosomal subunit, or a part of a ribosome. And coming in here, the big purple nose is another ribosomal subunit. And so this is actually for prokaryotic translation, but it doesn't matter really. The process is pretty much the same in eukaryotes. So this is the tRNA, which I'm circling right here. You'll notice that it has a sequence which complements the codon on the messenger RNA. So this is called the anticodon. And so that is how the tRNA recognizes that. Up here at the top is methionine. This is going to be the amino acid, so I want you to watch for those as they come in. So there are 3 sections on this ribosome that you're going to look at. There's the P site, where we're going to assemble the polypeptide. There's the A site, where each new tRNA is going to fly in. And then there's the E site, the exit or the eject, where you're going to eject a tRNA. So in comes another tRNA, and you'll see we have CCG as the codon, so the anticodon is going to be GGC. And now the ribosome is going to advance forward, and when it does that, you see the amino acid from the first tRNA is going to move to the other amino acid in the A site. The ribosome now advances 1 codon forward, and in is going to come a new tRNA matching the codon. This is UAC, which codes for tyrosine, and the ribosome is going to advance forward. Now if you pay attention over here on the left, in the E site, this tRNA is going to be ejected and fly off. It's going to go get another amino acid on it. It's going to continue to happen over and over and over again as we're reading the messenger RNA. And this is a very short one for demonstration purposes. And finally, we're going to arrive at a sequence which codes for a stop codon, which means the assembly is now done, and the ribosome is going to fall off of the messenger RNA, and translation is complete. We have a protein. There we go. And so what we're looking at here is a tRNA, and you can see down here would be, this is actually just, those tRNAs are a bunch of nucleotides. And so at the very bottom, the bottom three here at the bottom of this is an anticodon, and you can see it kind of looks like a T, so that maybe will help you remember it. And here would be at the 3' end where the amino acid would attach. The third codon tends to not be as important and allows for some tRNAs to bind to more than one codon. That's something that's probably not super important, so we'll come back to that later. This isn't too important here, so I'm going to skip that for now. And so here you can kind of see a quick recap of the animation, where we have the tRNA on the P site. We have another tRNA coming into the A site. It's going to be a little bit of energy that's required to assemble the polypeptide from one amino acid to the next. And now as that happens, you can see down here in diagram 3, the ribosome is going to advance forward, ejecting this tRNA over here in site 3, and the one in the A site moves to the P site. And all the while we're assembling our polypeptide. Remember, this occurs until we hit the codon sequence which codes for a stop codon. There are 3 of those. You don't need to memorize them. You'll be provided with a codon chart. This doesn't just happen with one ribosome at a time. There can be multiple ribosomes all attached to one messenger RNA. That's called the polyribosome. So this could be making multiple polypeptides at one time. So here's an electron microscope picture of that, all these different ribosomes attached to this single mRNA molecule. In addition to that, if we were in a prokaryote, this could also be happening in the cytosol, where we have multiple, this is in the blue line here is our messenger RNA. It's making the messenger RNA, which is this red line, and so while that is being made and moving along, you have ribosomes at the same time making that protein, allowing this process to happen really fast, and many different proteins being produced quickly. So to recap, if we're in a eukaryotic cell, this would, transcription would be happening in the nucleus. It then would leave after it gets processed and cut out all of our introns. And so here's our final mRNA. Then a ribosome would attach to it, and we would make our protein. So if Mr. Naveau could pause the video right now, I'd like you to answer these questions on a whiteboard, and in just a moment I will tell you the answers. Pause. Okay, so what is the end goal of translation? It is to make a protein. Number 1, make a protein or a polypeptide. Number 2, what form of RNA brings in the amino acids to assemble the protein? That would be tRNA. 3, what form of RNA facilitates the construction or synthesis of a protein? That would be the ribosome, which is called rRNA, ribosomal RNA. What happens at the A, P, and E site? At the A site is where the tRNAs first come in. At the P site is where the polypeptides are assembled. And at the E site, the tRNA is ejected or exits the ribosome. And lastly, what's happening on a polyribosome? Multiple ribosomes are producing a protein. And so there you have it. Continue on to the next video.